We all know about the amino acids, and we're going to start putting them together into proteins. So everything we've done up till now has just been reviewing, just setting the basics. So we're going to start putting things together into some heavy-duty biochemistry. So first we're going to study about my passion, proteins. Proteins are beautiful, beautiful molecules. First we're going to start to talk about the structure, and then we're going to talk about the function. We're going to spend some time on a very, very important class of proteins called enzymes. Now, our proteins are not colored like this, although that would be kind of cool. They're not necessarily colored like this, but this is colored. This is a pentamer. It has 5 subunits that come together to form the active molecule, the active protein. Now,
so that would be in a quaternary shape?
That would be the quaternary shape, that's right. You read ahead. Very good, Dylan. So here, this is a pentamer. This is actually C-reactive protein. Anybody in healthcare? Anybody? So where would you look for C-reactive protein? After what event in a patient? Yeah, some sort of trauma, heart attack. You're going to look for the C-reactive protein. Especially when you can't see the trauma, right? If you can't see the trauma, you're going to look for that. All right, so, sorry guys. To study the structure of our proteins, we're going to look at the levels. We're going to break it down into levels to simplify it. It's quite complex. So we'll look at the interactions involved, and this is where our review of general chemistry really comes into play. We're going to then look at some examples of protein structure, myoglobin and hemoglobin. We're going to talk about how biochemists determine the protein structure, and then how we can fold those proteins into our active structures. So again, why are we so worried about the structure of a protein? Remember, I said that the information in human life is held in the highly complex 3-dimensional structure of a protein. So where computers' information is zeros and ones, our information is in the 3-dimensional shape of our proteins. So all of the proteins' interactions depend on this 3-dimensional conformation. If the 3-dimensional conformation is incorrect, bad things can happen, and we'll talk about that. However, there's many conformations that are possible, but only one is native or active, biologically active. We call the biologically active form the native conformation. We can have really bad things happen if we do not fold it correctly. So the 3-dimensional shape that is biologically active is the native conformation. So to simplify the complexity of protein structure, we're going to break it down into 4 levels. First is what amino acid is bound to what amino acid. Okay, we call that the sequence of amino acids. That's the primary structure. What is the force held here? What joins an amino acid to an amino acid? Peptide bond. And peptide bond is also called an amide bond. An amide bond. All right, and we read it from the N terminus where you have the amine group free to the C terminus where you have the carboxyl group free. The secondary structure is a local 3-dimensional structure. We call them alpha helix and beta sheets, where it doesn't tell you the whole story about the 3-dimensional shape of the protein, but it tells you about a part of it. And these are common from protein to protein. A lot of proteins have alpha helix. A lot of proteins have beta sheets. Now, for most proteins, the tertiary structure is the whole story, how the protein is folded up. And it will give you that native conformation. So how are we going to fold up the secondary structure and the primary structure in order to get that active protein? And we're going to talk about our intermolecular forces that are going to be bringing those amino acid side chains together in order to have this three-dimensional structure. If we have more than one subunit, like our C-reactive protein, which is a pentamer, those subunits, those independent chains, are going to interact and form a quaternary structure. Not all proteins have quaternary structures. When we get to our examples, myoglobin only has one chain, so it's just a one unit protein, where hemoglobin has 4 subunits, so it has a quaternary structure. So myoglobin does not have a quaternary structure, but hemoglobin does. So not all proteins have this quaternary structure. Only if more than one subunit comes together does it have a quaternary structure. Let's see what this YouTube says. Internet's kind of slow this morning. Maybe it doesn't want to come up. Come on. We were told last week that the bandwidth was increased for this building. Hopefully there won't be a commercial. Come on.
Feel a spark of emotion light up every inch of you.
Let's
Feel warm inside. Feel our big, beautiful candle. Feel Blade.
S.C. Johnson.
Proteins are polymers constructed from the same set of 20 different amino acids. The primary structure is the specific sequence of amino acids specified by the gene. In the secondary structure, portions of the protein begin to coil and fold into unique three-dimensional formations. 2 kinds of secondary structure units are the alpha helix and the beta-pleated sheet. The folds and coils of the protein are the result of hydrogen bonds that occur at regular intervals. The tertiary structure is formed by interactions between side chains of various amino acids. At this stage, some proteins are complete. Other proteins incorporate multiple polypeptide subunits, creating what is known as the quaternary structure.
So I'm really impressed that there's a commercial for that one, because that means that it has enough hits to get, really? That's awesome. I don't know. I think that's really cool. All right, so enough people are interested in, maybe my YouTubes will get commercials. Actually, when I put this online, it will all be private. It won't be public. All right. So I like that video because it shows you how things come together. That primary structure, you have a covalent bond, right? The amide bond, the peptide bond, that is putting them, joining one amino acid to another, giving you the amino acid sequence. The secondary structure is just a local three-dimensional structure. And this is going to involve, and as we'll learn, hydrogen bonding in the peptide backbone. No R group interaction, no side chain interaction. We'll see how that comes together. We'll spend a lot of time today on that. Then we move on and fold up the entire protein, the entire polypeptide chain, into an active three-dimensional structure. Sometimes this is the entire story for a protein. But what we're having going on here is that the R groups, the side chains that we've studied in the last chapter, whether it be non-polar, polar, acidic, or basic, they interact in certain characteristic ways to fold up into our tertiary structure. If we do not have multiple subunits that interact, we are done with the structure of our protein. If we have subunits that come in to interact, like our myoglobin that we show here, we have a quaternary structure. And they're the same interactions that are involved in our tertiary structure. we define if a protein only has intact the primary or secondary structures, it's inactive, or we say it's denatured. The active protein must have its tertiary, or if it has a quaternary structure, intact in order for it to be active.
In the secondary, you said the hydrogen bonds, it doesn't bind here, or it doesn't?
No, and no R group interaction. No R groups. Christina, we're going to spend an awful lot of time on this, so we will beat it to death. So these are our native conformation. Anytime we have an active protein, we call it native. All right, so let's spend time on each. Again, that primary structure is the sequence amino acids. And it's very important to have this correct. All right? If we have a mutation, it can be pretty bad. A hemoglobin with a glutamic acid replaced with a valine in a beta chain makes that area more nonpolar because the valine only has carbons and hydrogens, where glutamic acid is charged. And so that's going to make that area more nonpolar, and that causes the protein to aggregate. And that gives us the aggregation of the hemoglobin gives us the sickle cell shape for sickle cell anemia. It distorts our red blood cell, giving that sickle's shape. It's not as elastic, so it can't deform as it passes through the capillaries. How long do normal cells survive? Does anybody know? Yeah, about 90 to 120 days, where a sickle cell anemia cell only lasts 10 to 20 days. So that's not good. That's not a good thing to have. And that's just one change in amino acid sequence, causing it to go from a charged amino acid to a nonpolar amino acid. Determination of the primary structure is very routine lab work. Chapter 5 is a chapter that I'm probably most expert on. However, that is a chapter we are not going to cover in this course. So if you're interested in it, I can sit down with you and talk to you for many hours about that. Hours and hours and hours. Or you can take my analytical chemistry class, which I talk about these things quite a bit. A little bit difference. There's some things that they cover in Chapter 5. You guys that took analytical, you'd find it interesting. Flip through it. There's some things that they talk about that we don't talk about, but maybe in other biotech classes they do. All right, so let's get into our secondary structure. When we refer to secondary structures, we are talking about structures that result from hydrogen bonding among the atoms in the peptide backbone. What do I mean by the peptide backbone? Right, let's look at our amino acid. It's going to go forward again on me. So this is the alpha carbon. Yeah. Oh, I'm going to erase that, sorry. Okay, I'm doing a little alpha. That's not a G there. That's alpha carbons, alpha carbons. This is our peptide backbone. I just drew the R groups as the R. And so what we have here, notice that we have a hydrogen bound to an electronegative atom, right? So hydrogen bonding can happen. And what can it interact with? it can interact with another electronegative atom. There's supposed to be two there. All right, so, but not right next to it. It might interact down the road. Okay? So we have interactions maybe with when it coils, as in our alpha helix. Here we would have, sorry guys, another carbonyl interacting. So this is our peptide backbone is what I drew out. Alpha carbon, that's a carbon, and I drew the alpha next to it. Peptide bond, alpha carbon. Oh, this is our carbonyl. And so on. Alpha carbon, peptide bond, alpha carbon, peptide bond. And so you're going to have that going on. That's your backbone. The R groups are not part of the backbone. So when I say the interactions of the backbone, I'm talking about that hydrogen that's bound to the nitrogen interacting with an oxygen and another carbonyl. Do you want me to say that again? The hydrogen on the peptide bond, on the nitrogen of the peptide bond, is going to hydrogen bond with the oxygen of a carbonyl down the road. Does everybody see that this is going to be a hydrogen bond? Now, along with that, we need to notice that there is free rotation around this alpha carbon. So here we have the alpha carbon. There's going to be free rotation on both sides, here and here. However, the amide bond does not. Okay? So it's going to make like a zigzag shape, and that's going to influence our shapes. So those factors, the factors of the free rotation around the alpha carbon and this hydrogen bonding that I've been talking about, results in two structures, two local structures, the alpha helix and beta sheet. So let's take a look at those. There's some information on these slides that I don't need you to remember, and I'm going to point them out. All right. I want you to take a look at this one right here. This is a nice one. I think it's in your textbook. So remember, the blue is a nitrogen, and this is a hydrogen. The reds are oxygens. So notice that the nitrogen, hydrogen, oxygen, carbon, this is that hydrogen bond. So you can see best on this one that it looks like a spiral staircase. You can imagine that the direction is the pole that goes up the middle. We don't have that. But imagine you have some braces on the outside of your spiral staircase. And what happens is that those braces are our hydrogen bonding. So here, it looks like you have a hydrogen here. They're not showing the hydrogen and the oxygen here. That's going to result in that hydrogen bonding. So the free rotation around the carbons, the alpha carbons, here and here. Oh, no, not there. Here and here, sorry. And this is the amide bond. That makes it conducive to forming our alpha helix. So I need you guys to know those two sort of parameters that result in the alpha helix and beta sheet. The free rotation around the alpha carbon and that hydrogen bonding. But I need you to be able to draw what that hydrogen bonding would be. So we have our chain, and we have a nitrogen covalently bound to a hydrogen. This is going to make this hydrogen susceptible to hydrogen bonding. Then, if you can imagine the chain wrapping around, and it coming near another carbonyl and another peptide bond, that's what we're going to interact. So I need you guys to be able to draw this. Can you see that, Emmanuel? Any questions about this hydrogen bonding in the backbone? This is for our secondary structure. So that's one factor that results. And the other factor that results in that alpha helix and beta sheet is this free rotation around the alpha carbon. There's no free rotation here. Now, a couple very interesting things happen as a result of this alpha helix and beta sheets. We'll talk about beta sheet next. But there's a couple very interesting things that happen. They're showing the R groups as the yellow groups up here. So take a look at the that one up there, okay, where I have the first arrow. Where are the R groups?
On the outside.
All on the outside. They're all on the outside of our alpha helix. So you can imagine our staircase, our spiral staircase, with the R groups on the outside ready to interact so that the alpha helix can fold up and interact with other alpha helix or other groups in our protein. I don't need you to remember that there's 3.6 amino acids per turn. I don't need that. They say that the alpha helix is clockwise or right-handed. So I have a hard time with this. So if you look at it, you take a look at the way it's turning. This is the direction. I think it's N terminus to C terminus. So the C terminus is going to go up to the ceiling. And then, and if you look at the turn, it goes this way. So imagine a staircase. You're at the top of the staircase. Either you're going to start walking down this way, or you're going to start walking down that way. It's really, I have a hard time imagining this, and I have a great imagination. So, but if you take a look at it, you can be either right-handed or left-handed. And the way I imagine it is being at the top of a spiral staircase, and which way am I going to start walking down it? That there's two different ways. I'm not going to ask you to remember which way or to draw it or anything like that, but just know that there's two ways that you can go when you have a coil. There's two ways you can do that. I do expect you to know that that peptide bond is trans in shape, okay, and planar. Trans meaning that the oxygen and the hydrogen are on our opposite sides. So we haven't said that before, that this peptide bond, the oxygen and the hydrogen are on the opposite sides of this bond that has double bond character. Remember that this
Why is it no free rotation?
Because it has double bond character. Yeah, it has double bond character. So really, it has no free rotation because there's double bond character. All right, and the oxygen and the nitrogen are on opposite sides. That's really important for our hydrogen bonds to be able to be set up. Now, it is interesting that this hydrogen and this oxygen, they're hydrogen bonding 4 amino acids away. I'm not going to test you on that, but there's 4 amino acids for our alpha helix. I do want you to know that if you imagine your spiral staircase, the hydrogen bonds go the same way that your staircase is moving. So you're moving up this way. And you put the pole in the middle, so that's the axis of our spiral staircase or alpha helix, that the hydrogen bonds go the same way. So imagine that you're growing your staircase. That's the axis of the alpha helix. Okay? Your hydrogen bonds are aligned with that pole in the middle. That means that it's parallel to the helical axis. Now this is the quaternary structure of tubulin, the protein I studied in grad school. I spent a lot of time with tubulin, as I've mentioned before. And it has 2 subunits that come together. And we're showing in the different colors the helical portions. This tubulin, this protein has a lot of helix structures. I don't think it has any beta sheets. And one thing I want you to notice is that you have a helical portion and then it stops. And it's just whatever. And then it starts up again. So this alpha helix is not the whole story. Or in the beta sheet when we get to that later, it's not the whole story. It can stop and then start again, and stop and start again. It's there when conditions are right for it. And then it stops when there's something that interrupts that. So let's talk about what interrupts that. Why would you have an alpha helix set up and then all of a sudden it stopped? We call those factors that disrupt that. All right, proline is one that stops it because if you remember, the structure of the R group actually comes back to the nitrogen. So here, the R group comes back in a cyclical structure, so we don't have that hydrogen. It binds itself to that nitrogen. So you can't have the hydrogen for the hydrogen bonding, and it causes no free rotation between that alpha carbon and the nitrogen. So because of that lack of free rotation, it disrupts the alpha helix. So that's for proline. So there's no hydrogen bonding. So you can't have the alpha helix if you have a proline. Remember, I said that on the outside of our alpha helix, the R groups are pointing out. If 2 R groups that are pointing out come close to each other that have similar charge, they're going to repel each other, and that can disrupt the alpha helix. So if you have 2 basic groups at physiological pH 7.4 are going to be positively charged. 2 acidic groups at physiological pH are going to be negatively charged. So if you have 2 negatively charged or 2 positively charged side groups that are close to each other, they are going to repel each other, and they're going to kind of travel away from each other, disrupt that nice. Yes.
So can we use fuse too? That's going to be able to repel?
I could.
It's going to be as few as like 2 amino acids that can do it.
That's right. It can be as few as 2 amino acids that cause a disruption in our alpha helix. Now, those of you that have taken organic with Professor Zubrick, this is known as the broccoli effect. And has anybody ever taken it with him? Okay. Did you take it with him? Yeah. So you heard the broccoli effect? Yeah. So the broccoli effect is when you have a big group. It takes up a lot of space. Imagine that valine, isoleucine, and threonine, they have big, bulky side chains. They're bigger. And can you imagine that they're so big that they're crowding out the other side chains, and they're pushing them away. So that can cause some crowding. Any questions so far? No? All right. Oh, yes,
You
Aaron.
said that last one, big who's pushing the side chains away.
Pushing other side chains away. So then that would stop the alpha helix from moving, from continuing. Now this is our beta pleated sheet. We have the same hydrogen bonding happening, but what we have is two sheets. Imagine my body is a amino acid chain, N terminus, C terminus, and it just goes through. All right? And what happens if they come together like this? I echo, but they come together like this. And then this is the nitrogen, hydrogen, and this is the oxygen. Look at that. That's coming together. They're hydrogen bonding, right? So that this makes the beta sheet. So you have two chains. They can either it can fold back, or it can go like this. Look at that. Is that weird? That can happen too. Here I had him. This is the what did I say? This is the N terminus. This is the C terminus. So they're going the opposite way, right? N terminus. Yeah, they're going the opposite way. Here, if I fold it back, they're going the same way. All right? And so here, because this is C terminus, this is C terminus, this is N terminus, that's the N terminus side. So here, they're interacting that way. So they can just fold back like this, or they can go like this. A lot of weird things can happen, and we'll talk about that later. But this is why I have the different arrows. So here, this top one was more like this, and this bottom one is more like when I go like this. So the basic point, what I need you to remember, is that you have two chains coming together. They can be on the same chain, but the parts of the chain come together, and they lie side by side. And we have the hydrogen bonding being set up. They can either be parallel, as we have in the top one, or anti-parallel, as we have in the bottom. But along the surface of the sheet, we have the hydrogen bonding.
So the top is parallel?
The top is the parallel, and the bottom is the anti-parallel. Now, there's a couple interesting things. This hydrogen bonding, we say, is perpendicular to the axis. So here, if you look at this way, the sheet is moving this way, right? But the hydrogen bonding is this way. So it's perpendicular to the axis of the sheet. What is super cool about this is that the R groups point up and then point down, and then point up and then point down, point up and down. So imagine a sheet. The R groups are lined up on the top and on the bottom of our sheet. Now, some weird things can happen with our beta pleated sheet. But before that, does anybody have any questions about how this comes together? So our secondary structure, we have the alpha helix, beta sheet. They both result from the same hydrogen bonding. and the same free rotation between the alpha carbon and the amine and the carboxyl group. So the alpha carbon on both sides has free rotation. See how it's that zigzag shape? There's no free rotation in the peptide bond, but then you have the hydrogen bonding occurring. If it occurs on a sheet perpendicular to the axis, it is a beta pleated sheet. If it's a coil and you have the hydrogen bonding happening on the outside of the coils, then you have the alpha helix. Again, with both, you have the R groups dangling, all right, ready to interact. With the alpha helix, they're on like spokes coming out, right? Imagine like hands hanging out of your spiral staircase, ready to slap someone else's hand. And then the alpha beta sheet, you have it pointing up and pointing down. All right, don't worry about that one. All right, so things that can happen to the beta sheet is this beta bulge. What happens is sometimes one amino acid is left out, where the length of one of the chains on one side of the beta sheet is longer than the other one. So that can result, I don't know what the difference between a wide bulge and a classic bulge is, but you can see that it's just one left out. One is left out. Here we leave one out, but then we have, this one is, it's left out, and this one is hydrogen bonding with something, sort of almost trying to do something else here. I don't expect you to memorize what's happening. I just want you to know that different things can happen. There's things that can happen where we stop the alpha helix or beta sheet. We get turns. Now, when you had the beta sheet, and say it happens like this, right where my neck is, right, there has to be a small amino acid, if it's on the inside, R group, in order to fit in this small area. Or if it's on the back of my neck, right, the R group's on the back there, it can be a big one. So glycine can be found in reverse turns because the R group for glycine is just hydrogen. So when it folds over, it's tiny. It can fit in there. Now, we also see proline in reverse turns. And then if there's a large R group, then it happens to be on the outside of that turn, it can cause that turn as well. Now, sometimes there's a little bit of fuzziness between our secondary and tertiary structures. Our secondary structures are only, and for this class we'll define it, and for exam purposes, I'm going to make it black and white. Secondary structures is only the interaction between the backbone of the peptide bond. And tertiary structures, you have interactions with the R group. But in reality, there's some fuzziness in between. So again, secondary structure, we only have the interaction of the backbone. You have that hydrogen bonding. And tertiary structure is only interaction of R groups. And that's what I'm going to test you on, but there are some exceptions, and I want to point it out. Sometimes we see a coming together of these alpha helix and beta sheets that happens over and over in many protein structures. We call those, when we see those over and over, we call them a super secondary structure. It's not the entire picture. A tertiary structure is the entire picture of that peptide chain. However, a super secondary structure is usually not the entire picture. It's like a part of a protein that comes together. And sometimes we see these, we call it beta, alpha, beta, where you have a beta sheet, an alpha helix, another beta sheet, an alpha, two helices come together. These are kind of strange. The beta meander and the Greek key is just really weird. I'm not going to test you on giving me the definition of these. I just want you to know that there is some fuzziness between those, and you do see these patterns occur over and over. And then there's another beta barrel. All right, so here the arrows are showing a beta sheet. And of course, the helix is the helix. So we will see these structures over and over in many of our proteins. So that's why we say, oh, yeah, that's the beta alpha beta portion of this protein. You know, and here's a beta meander. When it's like jargon talking about things on a protein structure. This is kind of weird. Beta sheet stops, beta sheet stops, beta sheet stops, beta sheet stops, beta sheet stops. So that's our beta meander. And then this is super cool. This is our Greek key. So sometimes I just stop and say, nature did this. Like on a molecular level, nature made this pattern that ancient Greece did. So it's almost like artists on the molecular level. So here you start out with a beta sheet, and then you make a little like sort of beta meander in the middle, and then when you're done with the beta meander in the middle, you come out and do another beta sheet. This happens enough in our protein structures that we name it the Greek key, which looks like this structure on this ancient Greek vase. Is anybody weirded out by that? I just think that that's kind of a strange thing to have happen in your protein structure. Sometimes I just stop and say, wow, that's a strange thing. So, and personally, my art in my life is protein structure. Like I think that that's nature's art. People might like flowers, people might like, fine art. I like protein structures. I find those to be beautiful because that, to me, is the basis of life. That the three-dimensional structure and the way that things fit together, that's the beauty of life. I'm sorry, I'm very philosophical today, but this is why I am here, all right? This is why I teach this class, and I find this very interesting. And once I started learning about proteins, I never wanted to stop learning about proteins. Because we can not only understand what life is about, but we can then use them to solve all sorts of problems. The company I worked for before I was here used proteins to solve problems and make a lot of money, too. So, all right. One protein that super secondary structure does give the entire story is collagen. Collagen has 3 chains that have a helical structure. It's not an alpha helix, but it is a helical structure. We're not going to get into it too much. And those 3 helical structures wrap around each other. So it is where the super secondary structure, the secondary structure sort of really does tell the whole story. And what does collagen do for us? And why do we get injections of it? I don't. Not yet. I should have, right? Think about it. Proline and hydroxyproline. Well, remember, hydroxyproline has an extra OH, and and hydroxylysine, those will attract water. So that's why people get collagen injections in their, is that what they put in those ladies' lips? Is it collagen? So that they have these enormous lips? Because it attracts water. So it makes them nice and plump and weird looking. That's not a good look. I don't know. In my book. Anyway, so collagen, we'll get this. Now, meat that is tough usually has a cross-linking between these strands. So that's what happens when meat gets tough. You'll get cross-linking there. And old people. Well, this is what happens in old people. I am one of them. All right, so we're getting pretty complex for secondary structure, right? So this is why I said there's a fuzziness between the secondary and tertiary structure. With age, they cross-linked with covalent bonds. All right. Tertiary structure. This is where all the beauty comes in. This is our whole story. Arrangement in space of all atoms in our chain. Again, it's not always clear where secondary ends and tertiary begins, but I'm going to make it clear for this class. Secondary ends when we're only talking about hydrogen bonding in the backbone, and tertiary begins when we talk about interactions of R groups, of our side chains. Those are groups that we classified in the last exam as polar, nonpolar, acidic, or basic. Quaternary is much the same as tertiary if we have different subunits that come together, the same interactions. Now, based on their three-dimensional structure, we have two types of proteins based on the shape that results in their tertiary structure. A fibrous protein that has the shape of a fiber, and globular proteins that have the shape of a glob. All right, let's talk about fibers first because they don't really do much other than give mechanical strength. They're long fibers or sheets. They're quite strong, and they usually are insoluble in water. So because of this, they're structural in nature. Keratin and wool, collagen of connective tissue in animals. So I think that I need to take this little treatment. But now you can get shampoos with it that are supposed to do that. I don't know if it would ever. She needs to stop dyeing her hair. That's my advice to her. That's a problem. But I think a few years ago you could go to the hairdresser, right, and get a keratin treatment. Would it really do this to your hair? Would it make that look like this? Styling does that to you. I don't know if that would do this to Dolly. But that's the keratin, and obviously it gives mechanical strength. It's fiber. All right, but globular ones, that is what we're very interested in. Again, here's my structure of protein tubulin. This is where a anti-cancer drug would bind. However, notice the shape is not really spherical. That's why we call it globular. More or less spherical. No, not really. Just a glob. All right. They tend to be soluble in water. Not always. The bigger they get, the less soluble they are. But what happens is, because they're soluble in water, mostly, that we're going to fold it, and so the nonpolar side chains are buried inside. And this is an entropy-driven force, and this is the strongest force or the strongest drive in folding of proteins, this entropy, trying to get rid of the nonpolar waxy side chains from the aqueous environment. We don't want to have that ordered chain around those nonpolar side chains. We want to have those buried inside. But there are a lot of interactions that come together when we are folding our proteins. Notice with tubulin you have, here's an alpha helix. Oh, there are some beta sheets. Some beta sheets thrown in there. So, but then it stops. When you see the strings, that's when there's no secondary structure there. All right, so you have an alpha helix, and for some reason it stops. And then there's a beta sheet. It looks like maybe there's a beta meander or something going on there. And, so it really does come along and form different groups. But I do want to point out that you do have portions of your protein that aren't involved in beta helixes or alpha beta sheets or alpha helixes. Most proteins do have some, but it's not the whole story. So here's a nice comparison of our fibrous and globular protein. globular, looks like, again, a glob. But remember, it does have this three-dimensional shape. So we're going to spend some time here talking about the forces. And again, as you're condensing your notes here, what I would do is go through the levels of your protein structure, define them, and really talk about the forces involved in each. So again, let's summarize. The primary structure, what's the force in the primary structure? Peptide bond. Secondary structure, you have alpha helix and beta sheet, and the force is the hydrogen bonding in the peptide bond. So in the backbone, in the peptide backbone. So when you define, if you don't say hydrogen bond in the peptide backbone, points off, right? We don't want points off for that. So, and then the tertiary structure, we're looking at the interaction between the side chains. So that's important. Now, see, the reason why in fibrous proteins the secondary structure will give us most of the story is because we don't really have a folding. It's fibrous. It's one long line. So the secondary structure does give most of the tertiary structure information. So we're going to see how things fold back on each other in a globular protein. All right, so the forces that are involved, and you got to know all of these. This is all side chain interactions. So let's think about our side chains. You might want to even look back in your notes on where I looked at those R groups, the 20 amino acids. Serine and threonine both have a hydroxy group. So they would have hydrogen bonding between each other. There's other ones that will have hydrogen bonding as well, but these are just examples. You have hydrophobic interactions between nonpolar side chains. Valine and isoleucine have nonpolar side chains. So those are going to interact. Why is that jumping? You guys see that jumping, or is it just me? It's jumping. Maybe people are walking around upstairs. Now, this is, we have electrostatic attraction between side chains of opposite charge. So if you have a basic one that's going to be positively charged at neutral pH or physiological pH, and an acidic one, like glutamic acid, that's going to be negatively charged at physiological pH, then those are going to form what we call a salt bridge. So there's a couple things I want you to know. Let's stick at this electrostatic attraction for a second. Our basic side chains are going to have a higher pKa.
Yeah.
And because of that higher pKa, at pH 7.4 is the pH of blood. What's the pH inside our cell? About the same, right? Somewhere around neutral, right? It's going to be positively charged. So below the pKa for our basic side chain, it's going to have proton on, which is going to make that nitrogen a quaternary amine, and it's going to be a positive charge. So summarize, basic side chains, positive charge. In cellular conditions. Acidic side chains have a low pKa. So when we're at neutral pH, that we're going to be above the pKa, and the proton will be off. So it will be negatively charged. So if we have a positively charged basic side chain and a negatively charged acidic side chain, they're going to be attracted, cation to anion. Now, if we put these acidic and basic side chains on the outside of our protein, what's going to surround the positive and the negative? Water molecules. Around the positive, the oxygen of the water molecule is going to surround it. They're going to be hydrated. Around the acidic side chain, the positive side of water is going to be around it. So where do you see these electrostatic attractions between the side chains of opposite charge? Are they going to be out on the outside of our protein or on the inside? On the inside. Has to be away from the water for salt bridges. So this another term for this electrostatic attraction between side chains of opposite charge, that's called the salt bridge. You're going to hear me talking about that. Salt bridge. Now, similarly, if you have two basic side chains that are positively charged or two acidic side chains that are negatively charged together, they're going to repel each other. So they're going to try to stay away from each other. Then there's one covalent interaction, and that's where we are going to oxidize two sulfurs on the two different cysteine residues. An amino acid, one amino acid, we call it amino acid residue. I don't know why. It's an amino acid residues. They are going to oxidize. We're going to get rid of the two hydrogens, and there's going to be a covalent bond. So two cysteines can come together and form a covalent bond. Did anybody ever do a gel electrophoresis? You know, did you use beta-mercaptoethanol, some smelly stuff? No, they don't do that anymore, I don't think. Okay, so you're going to have to add, when you do SDS, gel electrophoresis, what you do is you add soap to it, and you also have to break these disulfide bonds. And so that is breaking. When you have a perm, what you do is you put the curlers in your hair, and then you break all of the disulfide bonds in your hair, and then you add another chemical to add them back in. But it adds it back in the form that the curler is in. I'm a child of the 80s, so I'm very familiar with perms. Men would often get perms with very long hair in the 80s. The music was great in the 80s, but the fashion was not. I look back and it's...
Bell bottoms are great, though.
Bell bottoms were not part of the 80s.
Well, that was the 70s.
That was the 70s.
What about parachute pants?
It was high waters, actually, in the 80s. We actually would roll up our pants. It was not a good thing. So you guys need to know all of those. But here's a nice little summary. So here, this is our alpha helix and our beta sheet. I don't know why they do this as a ladder. These are supposed to be the red dots, just like we showed the hydrogen bonds here. So that's not it. But these are not the tertiary structure. But we are going to look at the interactions between these. So here, we're showing that hydrophobic interaction, where our nonpolar side chains are going to go into a little pocket. Here's some hydrogen bonding between a hydroxy group and maybe an acidic group. Doesn't really matter. Anything that can do hydrogen bonding. Here's a salt bridge between a positively charged basic side chain and a negatively charged acidic side chain. Here's our disulfide bond between two cysteine amino acids. We can have some metal coordination we'll talk about later. But here's, it's just a nice little example. What time is it? Yeah, 9:30. So I'm going to go through a couple more slides, and then we'll stop for today. So in our quest to understand how proteins more, we often want to know the exact three-dimensional shape, because we can't just take a look at the amino acid sequence and say, hey, it's going to fold like that. We can take a guess, and we'll talk about that later, but we don't know for sure. So we take an X-ray of our protein. Now, there's some downsides to this, that you have to have a protein that's a crystal, that's a solid. When proteins are native, they're in an aqueous situation. So not always are we going to be able to use a crystal. And this is, when I was in grad school, a big bone of contention in the tubulin world, where someone had come up with an X-ray of tubulin and said, hey, this is our tertiary structure, where we know tubulin was big. If it was in a solid form, it was not going to be in its native form. So they came out with this structure, and my group that I was researching with disagreed with them. We said, we don't believe that when you make a crystal of tubulin that it's going to be in its native conformation. But it is an X-ray, but what happens is because it's on a molecular level, what happens is they pass the X-rays through, and based on the interactions of light, remember when you pass light through and it comes back and it forms diffraction pattern? You get that diffraction pattern, and you have to do a Fourier transform on it, and you can't do it. You get some, and I'll show it to you in the next page, but you need a computer to get the information off of it. What is more modern is to do a 2D NMR, and those of you that have come from RPI, they have NMR that is strong enough to be able to do it. In fact, I used to take my analytical students over to RPI to see the NMR instruments that you would have to climb on a ladder in order to go down and put the sample into it. Anybody come from RPI? Anybody graduate from RPI? No. But my contact there did not get tenure because he was too good of a teacher. I would bring my students over, and he would explain NMR in a way that I've never heard in such simplicity. I could never repeat. He was so good. But because he was too good of a teacher, they had to get rid of him. So he no longer is there, so I don't bring my students there anymore. But NMR looks at the chemical environment around carbons and hydrogens, for those of you who have never heard of that before. But what I like about it is that it can be in its natural environment in aqueous solutions. So here's the diffraction pattern that gives us the tertiary structure of a crystal, and this is 2D NMR. From this, we can look at the structure of our protein. I know that's very complicated, but when this is your life's work, you will get some information out of this. But yeah, this is wicked hard to do. One-dimensional NMR is hard enough for those of you that had organic. Two-dimensional, yeah, I got into in grad school, but this is with proteins. You really have to have an expert helping you out. All right. I think I'm going to stop there to give back your exam and take attendance. Actually, let me take attendance first. But let me